Good afternoon, students.
Good afternoon, teacher.
How are you?
We are fine, thank you. And you?
Thanks, I am fine. Sit down. In the last lesson, we studied the structure and function of cell. Today we continue explore the function and structure of cell. We know there are two types of cell: single membrane cell, two membrane cell, and one membrane cell. Today we study the single membrane cell. Today we have lesson objective. Explain the features of structure and functions of a cell, organelles visible under the electron microscope. Explain the new terms: tanks, proximal site, distal site, smooth endoplasmic reticulum, rough endoplasmic reticulum, and attached. You are divided into 3 groups when listing my instruction. The first group, you are prepared information for endoplasmic reticulum, about structure and function of endoplasmic reticulum using the mental map strategy. Thus, the second group show information for Golgi apparatus and lysosomes, and the third group make the cluster using the word association. You have 4 assessment criteria. Your information should carry content, use of your new terms, don't forget, keep time, and after your presentation I have few questions or tasks. For each criterion you have 1 point. And please, the first group, your presentation.
Endoplasmic reticulum is a single membrane organoid. This is system on the tanks and channels connected to all the and limiting the single membrane of the cavity of the X.
A membrane is single connected with the one nuclear membrane.
There are 2 types of AR, raw, otherwise called granular, and smooth and agranular.
Smooth AR synthesize carbohydrates and lipids, and on the left AR we have a ribosome that synthesize proteins.
APR transport substances. Protein molecules are transported to the APR channels to the Golgi apparatus.
I agree with you. Excellent. I am fine. That's right. I really enjoyed it. The first group, good job. And sit down and perform the second group. The second group shows the performance about the Golgi apparatus and lysosomes. Please.
I'm a Golgi apparatus. Golgi consists of tanks with a bubble system. There are many bubbles on site attached to the ER. They transfer lipids and proteins from ER to the Golgi. The most important function of Golgi is elimination of various secrets such as enzymes and hormones from the cell. Golgi is well developed in secretory cells. It's called the cell export system. Therefore, Golgi has 2 sites.
I am the proximal site. I give from APR, lipids and carbohydrates.
I'm a distal site. My nature site has inside proteins, bubbles, carbohydrates and lipids. These bubbles go beyond the Golgi to the destination, and beyond this substance form the lysosomes.
I am primary lysosome. I can contain from 20 to 60 different types of hydrolytic enzymes. Lysosomes, which are bodied the apparatus Golgi, are called primary.
Autophagy is the process of destroying unnecessary cell structures. First, the structure to be destroyed is surrounded by a single membrane. Then the formed membrane vesicle merges with the primary lysosome. As a result, the secondary lysosome also forms an autophagous vacuole in which this structure is digested.
The second group make good job, sit down, and presentation the third group. Third group make cluster using the word association. Please, third group, vacuole.
We made clusters using the word association. Vacuoles. Vacuoles are the single membrane organoids. Vacuoles are containers filled with aqueous solutions of organic and inorganic substances. EPR and Golgi apparatus take part in the formation of vacuoles.
The membrane limiting vacuole is named cytoplasm.
The liquid that fills vegetation is called cell juice.
In animal cells, there are small digestive and aftervacuous vacuoles. In unicellular animals, there are contractile vacuoles that perform the function of osmoregulation and excretion.
Plant vacuoles are responsible for the accumulation of water and the maintenance of turgor pressure. The accumulation of water-soluble metabolites reserve nutrients and mineral salts. The coloring of flowers and fruits, there will be the conversion of pollinators and seed distributors.
The all groups performs very well. I have any questions and tasks for each groups. The first groups, you see the food contains the protein, carbohydrates and lipids. What is substance? Synthesis on roof endoplasmic reticulum was substance synthesis of smooth endoplasmic reticulum. Nurdana?
Okay, roof endoplasmic reticulum, we choose, our group choose dairy products and eggs because they consist on protein and therefore systemated in roof endoplasmic reticulum.
I agree with you, Nurdana. That's right, sit down. And smooth endoplasmic reticulum. What food contain the substance synthesis of roof endoplasmic reticulum? Nazirke?
With smooth endoplasmic reticulum, I connect the rice, nuts, potato, and the oil. Rice and potato, they have carbohydrates, and the oil, nuts, they have more lipids.
That's right, Nazirke. This food have more carbohydrates and lipids, whereas synthesis on the smooth endoplasmic reticulum. The third group, I have your few questions and tasks. This plant has aileron grains. What is plants? Karima?
I think it's wheat and potato because when vacuole is dehydrated, they turn to aileron grains.
I agree with you. That's right. Potato and wheat has aileron grains. The vacuole juice modifications for aileron grains. That's right. Make the feedback. The all groups performs very well, worked very well, and speaker of first, second, third group. What your was opinions for the all groups? Whom said the opinion? Aray.
Our group really liked work of first and third groups. Their explanation and clusters were really interesting. We gave for each group for about 4 points.
Do you understand everything?
Yes.
Yes. I give the homework, paragraph 12 and exercise 4 and rewrite the new terms. Good luck!